Hello, my name is Riley Black, and the topic I chose to research for this project is should the U.S. allow the use of human germline engineering to correct monogenic disorders? Alright, starting off with my background, the first genome editing technology was introduced in 1984 with successful insertion of genetic material into mice. This was the starting point for the possibility of gene editing. There are currently two main techniques used, the first being germline engineering, which alters the genome of a human embryo at its earliest stages. This may affect every cell, which means it has an impact on not only the individual, but on his or her descendants as well. The second way is through somatic cell engineering. This involves modifying a patient's DNA to treat or cure a disease caused by a genetic mutation. And as you can see by this photo, it differentiates somatic gene editing versus germline gene editing. In order to correct monogenic disorders through the alteration of the human germline, CRISPR-Cas9 is the gene editing technology used and is what I will be referencing throughout this presentation. It involves two essential components, a guide RNA to match a desired target gene, and Cas9, which is an endonuclease, which causes a double-stranded DNA break, allowing modifications to the genome. In many studies, it has been proposed to limit germline editing to severe cases of disease and disability where no alternative treatment is possible. For example, there are more than 780 monogenic neuromuscular diseases linked to 417 different human genes. The CRISPR-Cas9 system has been used to remove the genetic defect, and I will be talking about how human germline engineering is most commonly used in the context of disease prevention and infertility treatment. I will now move into my pro arguments. My first argument is that engineering the human germline has the ability to reduce the prevalence of monogenic inherited diseases. CRISPR-Cas9 allows for precise modifications in the genome by creating double-stranded breaks at specific locations. This uses the application of CRISPR in human germline cells, including pre-implantation embryos in male and female germ cells. Correction of the mutations in the germline could allow the patients to produce sperm or oocytes that are free from the mutation, therefore producing healthy, mutation-free embryos. In August of 2017, the first therapeutic germline intervention using CRISPR-Cas9 was reported. A team from Oregon Health and Science University created zygotes by fertilizing healthy oocytes with sperm cells from a carrier of the MYBPC3 mutation. This mutation leads to heterotrophic cardiomyopathy. By using CRISPR-Cas9, the team corrected genetic defects in the zygotes, which then led to the development of viable embryos. Since this was used for research purposes, the embryos were not implanted into a female uterus. However, their research shows that correcting a gene mutation in viable human embryos using genome editing methods is in fact feasible. My second pro is that correcting monogenic disorders improves one's quality of life. An article published in Clinical Genetics talks about how gene editing technologies are expected to significantly improve the quality of life for those suffering from genetic disorders. Since monogenic diseases are related to mutations in one specific gene, engineering holds promise in the future of correcting genetic diseases such as sickle cell anemia, cystic fibrosis, Huntington's disease, and many more. These conditions have the ability to reduce life expectancy by decades. By editing the human germline, it may soon be possible to prevent these diseases from being passed on to offspring, therefore ensuring better quality of life. My third and final pro is that the edited gene is copied in every cell and passed on to future generations, eliminating diseases in certain lineages. When a gene is engineered in the human germline, whether that is sperm, egg, or early embryos, the genetic modification can be passed down generation after generation. It is approximated that monogenic diseases account for 10 in every 1,000 births. Changes made in the DNA are incorporated into the DNA of the resulting embryos. The genetic alteration is inherited in the same way as any naturally occurring genetic trait, effectively eliminating or correcting the inherited disease in future generations. The CRISPR-Cas9 gene editing system is the first technology that allows for efficient modification of DNA sequences. Using germline editing to study early development, this has the ability to give us a better understanding of the causes of infertility, therefore providing better treatment options and a new way to study human development. To summarize my pro-arguments, germline gene editing using CRISPR-Cas9 offers a promising approach to reducing the prevalence of monogenic inherited diseases by enabling precise genetic modifications in embryos and reproductive cells. As previously stated, the 2017 researchers successfully corrected the MYBPC3 mutation in human zygotes, demonstrating that this technology is feasible. Beyond preventing inherited diseases, gene editing has the potential to significantly improve quality of life, eliminating conditions such as sickle cell anemia and cystic fibrosis, which can drastically reduce life expectancy. Studying early development through germline editing may lead to a better understanding of infertility and improved reproductive treatments. Now on to my con arguments. My first con is how there are unintended genetic consequences such as point mutations and genetic mosaicism. Human gene engineering comes with practical challenges such as off-target mutagenesis, mosaicism, and large deletions and rearrangements. CRISPR-Cas9 can cause unintended mutations at genomic locations with similar sequences. While some studies have reported minimal off-target effects in human embryos, others have shown substantial off-target effects specifically within mouse models. The risk of off-target mutagenesis is dependent upon the target sequence and needs additional research to perfect. Additionally, mosaicism occurs when different cells in an embryo have varying genetic modifications due to the timing of the CRISPR-Cas9 activity. This can result in organisms with mixed genotypes rather than eliminating a monogenic disorder. Human gene engineering can also cause unintended large deletions or genomic rearrangements, leading to gene loss or misregulation. These are hard to detect with conventional genotyping methods, making it difficult to assess the risks of gene editing. Some studies suggest that large-scale deletions and rearrangements are more common than initially thought, and these mutations need to be better understood and detectable. My second con is that several ASO drugs have received FDA approval for treating monogenic disorders. However, their ineffective delivery remains an obstacle in research. ASOs are short, synthetic DNA or RNA sequences designed to bind to specific RNA molecules to modify gene expression. For ASOs to reach their target site within cells at effective concentrations, they must overcome several biological barriers. These include resisting degradation by extracellular nucleases, evading phagocytic clearance, and crossing the capillary endothelium. Because severe threats to the entire human population may arise as the result of uncontrolled genetic manipulation, it is essential to prove effective delivery in many trials. My last con is how there is a lack of research and regulation associated with altering the human germline. Germline editing is banned in the United States by acts of Congress. However, there is no law or regulation that bans germline gene engineering conducted through private funding. Risk assessment must be performed case by case to ensure the safety of each specific gene that is being engineered in the human germline. Because severe threats to the entire population may arise, regulation must be in place. As of 2014, approximately 40 countries, including 15 in Western Europe, had discouraged or banned research on germline editing due to safety concerns. And here on this slide, I have a pie chart that depicts policies that are in place on human germline genome editing in 96 different countries. So you can see that there's not a whole lot of relevant information, or when there is, editing the human germline is prohibited. To summarize my con arguments, germline gene editing presents significant risks due to the unintended genetic consequences, which can lead to gene misregulation or loss entirely. While some studies report minimal off-target effects, others have shown substantial unintended mutations, highlighting the need for further research. And like I mentioned previously, although ASO drugs have received approval for treating some disorders, their ineffective delivery still remains a big challenge. The lack of research and regulation further complicates concerns surrounding germline editing, and it also comes with the high risk of the creation of designer babies, as well as a high cost for healthcare companies. If this technology becomes widely available, it could create a society where genetic modifications and designer babies are the new standard of care. Without comprehensive oversight and proven safety measures, germline editing remains a highly controversial and uncertain scientific endeavor. So with that, I hope you have a better understanding of my topic, and I want you all to leave your thinking. Do you think that the U.S. should allow the use of human germline engineering to correct these disorders, specifically through the CRISPR-Cas9 technology? This topic will need to be carefully considered and regulated, but there is a lot of potential for future medical advancements. This concludes my presentation on whether or not the United States should allow the use of human germline editing to correct monogenic disorders. So thank you all for listening.